All right, let's dive in. Today, we're talking about something pretty wild, hacking the actual code of life. We're getting into systems biology, a totally revolutionary field that is completely changing how we understand everything, from a single cell all the way up to the most complex diseases.
So, to really wrap our heads around this, it all starts with one simple but really fundamental question, one that kind of frames this entire new way of looking at biology.
I mean, think about it this way. Imagine a huge city. Are you going to learn more about how that city really works by just studying one single traffic light? Or by looking at the entire traffic grid, the power lines, the water systems, and seeing how they all connect and interact? That right there, that's the core idea. And this right here is the perfect way to see that shift in thinking. The old way, well, it was what we call reductionist. You'd focus on one tiny piece, like one single building in our city. But the new way, the systems way, is holistic. It's all about understanding that entire interacting network, the whole city grid. You know, for decades, biologists did some absolutely incredible work studying just one gene or one protein at a time. But they started to realize that inside the insanely complex city of a living cell, everything is connected. Studying one part in isolation, it just wasn't giving them the whole story. So, to tackle this incredible complexity head-on, a whole new field had to be born. One that was designed from the ground up to handle all of those moving parts at the exact same time. And here it is. So, what does that actually mean? Well, systems biology isn't just about collecting a ton of data. The key word here is integrating it. The real goal is to build a working, computational model so we can see how all those different parts, the genes, the proteins, and other molecules, are all talking to each other. And you know, this wasn't some overnight revolution. The seeds were planted way back in the 50s and 60s with some of the first computer models of things like nerve cells and the heart. But things really took off in the 2000s, especially with the founding of the Institute for Systems Biology. And then came the big one. In 2012, we got the first ever complete computer model of an entire living organism. It was a tiny bacterium, sure, but wow, what a huge leap forward. Okay, so how on earth do you even start to build a map of something as complex as a cell? Well, you need data, lots of it. And it comes in these different layers that scientists call the omics. First up, you've got the bedrock, genomics. This is the study of an organism's complete set of DNA, its genome. Think of it as the entire genetic library, containing every single instruction the organism could ever possibly need to build and run itself. So going back to our city metaphor, genomics is basically the master blueprint. It's got the plans for every single building, every road, every park that could ever possibly exist in that city. It is the ultimate instruction manual. But here's the thing. A city doesn't build everything all at once, right? That's where the next layer comes in, transcriptomics. By measuring all of the RNA in a cell, we can see which of those blueprints, which genes, are actually being used right now. It's a snapshot of what the cell is actually doing. So, in our city, it's like we're flying over it at night. We can't see the whole blueprint from up there, but we can see exactly which buildings have their lights on. It tells us what's active and what's happening at this very moment. It's a dynamic, living picture.
And finally, this is where the action happens, proteomics. If genes are the blueprints, then proteins are the city's entire workforce. They're the builders, the messengers, the power generators. They are the actual workers and machines that carry out the instructions and make everything run.
And this is what really brings the whole picture to life. Proteomics takes us inside those buildings that have their lights on. It's not just about knowing a gene is active. It's about seeing the people and the machines, the proteins, actually doing the work. This is the action layer, where biology truly happens. Okay, so you've got these absolute mountains of data from genomics, transcriptomics, and proteomics. We're talking terabytes of information. How in the world do you turn all that raw data into something you can actually understand? Well, that requires a very specific and very powerful set of tools. You need a seriously powerful toolkit. And this is where systems biology gets really cool, because it's not just biology anymore. It's this mashup of biology, computer science, and math. Scientists use bioinformatics to organize all the data. Then, they build computational models to simulate how the cell behaves. And then, they use network analysis to map out all those incredibly intricate connections. And scientists have basically two main ways of attacking this problem. There's the bottom-up approach, where you start with small experiments on individual parts and you build the model piece by piece. You go from experiment to model. Then there's the top-down approach, where you start with huge data sets to predict a model first, and then you run experiments to see if you are right. Both are super important. All right, this all sounds really cool in a lab, but what about the real world? Where does this actually make a difference? Well, one of the biggest arenas is the fight against cancer. See, the key insight here is that cancer isn't just one broken gene. It's the whole network going haywire. And if we can map that entire broken network, we can find its unique weak spots and figure out the smartest way to shut it down.
So here's how they do it in the real world. First, scientists get a complete omics profile of a person's tumor. They look at its DNA, its active genes, even its epigenetics, which are kind of like sticky notes on the DNA that tell genes to turn on or off. Then they put all that data together to build a model of that specific cancer's network. And that lets them see exactly which pathways are broken and, this is the most important part, find totally new targets for drugs.
And this is the whole point. This is the key to what we call personalized medicine. Instead of just giving everyone the same treatment, doctors can actually design a therapy that's tailor-made to shut down the specific network that's driving one person's individual cancer. It's a game changer.
And believe me, the applications in cancer are just the beginning. This way of thinking is blowing the doors wide open for science and medicine.
I mean, just think about what's on the horizon. We're talking about designing completely new drugs and vaccines, predicting how entire ecosystems will respond to climate change, engineering brand new biological functions that don't even exist in nature, and maybe even building a synthetic living cell completely from scratch. Which really leaves us with this one kind of mind-blowing question. For centuries, we've just been observers of life's complexity. Now, for the first time, we're actually starting to read and understand the instruction manual. So, are we getting ready to write a few pages of our own?